Keith, do you remember how many are supposed to be here?
We had just a very few more than this on this morning, in the low 50s somewhere. So I think
Well,
probably
we can wait a couple
wait just a
minutes.
little bit and then go ahead and take off. And
Yeah,
good
no
to see everybody.
worries.
Hope you all found some nice lunch.
Or at least got up to walk around a little
Yeah,
bit and give
some
yourself
change
a
of pace.
break. Yeah. I think my mouse may be dying. There. Yeah, probably. Okay. So is it all bringing back memories from last year? Some of the material, hopefully not so out of usual. All right. Well, and the good thing is that we are recording, so you will have access to this afterwards for studying. And now that I'm in the dark because my light doesn't recognize the fact that I'm actually sitting here moving around, but it's not worth me getting up and doing it because it'll just go off again. All right, let's go ahead and get started. I'll go ahead and share screen again so that you see the where we'll pick up where we left off. Okay. So we finished talking about the consequences of variation, and we had two examples and two different types of transporters that we went over. But now, one of the things that is the case most of the time is that it's not a simple one-to-one relationship where a polymorphism in one gene ends up giving you a result and it's a clean result, okay? Because as we all know, biology is a whole lot more complex than that. So the examples here are of a monogenic trait on the left and a multigenic trait on the right. And so if we have, I better give up with that mouse, turn it off, so I'm not tempted to use it. So if we have a monogenic trait and we've got a polymorphisms, two potential alleles, one with low activity and one with high, you can imagine that if we have a case where a certain segment of the population is homozygous for the low activity allele, then those folks are going to have low activity, okay? Because they don't have the potential for even having a high activity allele. And so if you have a segment of the population who's homozygous for the high activity allele, that means they have two copies of that high activity allele. You're going to end up with those folks having very high enzyme activity as a portion of the population. And the cool thing about enzymes is that you can have a mixture. So if you have a allele that encodes for low activity and high activity, so this person is heterozygous for that in that gene, now you're going to end up having enzyme activity that's somewhere in the middle. Okay, makes sense. You've got half of your alleles, potentially half of the alleles that that encode these low activity enzymes, and the other half are high activity. Well, that's all well and good in the simple things, but what happens if you have multiple genes that are influencing this one activity, right? That's called a multigenic trait. We talked about that a little bit. So if, once again, you have alleles with low and high activity. And in this case, we're looking at a histogram for multigenic traits. And you can see all the various combinations here. So if you have the low activity allele for 2, you have 2A, and then you have one of the 3As. Now, all of a sudden, you're going to have really low activity. So you're going to have a lower thrombosis risk in this scheme of things. But depending upon which combinations you have, you have 2A, and then you have 3C. 3C is actually missing a whole exon here. Then you're going to have something a little bit more risk of thrombosis. Once again, here you go. What happens if you have, 4A, you're heterozygous for 4, and then you have all of these other combinations? You're going to have higher and higher thrombosis risk. So this is just kind of an example of how, when you're looking at the combination of 4 different genes and 2, 4, 6, 8, 10, 11 different alleles and 11 different possibilities here, you can imagine how complex it can be. And that's why you're not going to have this nice clean histogram of either low, medium, or high activity. In the population, the thrombosis risk is going to have this histogram as a unimodal skewed result. And you're not going to have those distinct clean differences between the different groups, genotype groups. So something I can't remember if we talked a lot about it or not, but there are multiple databases that contain information on the human genetic variation. PharmGKB, Pharmacogenomics Knowledge Base, is one of the biggest and one of the best. DBSNP used to be really well curated, not as much anymore. GWAS, if you remember what GWAS stood for, Genome-Wide Association Studies, there is a database for those, and they're really good at describing genotype-phenotype associations. So one that I emphasized for you guys quite a bit was online Mendelian inheritance in man, OMIM. And I have to tell you, I just had a P4 who was on their API this summer and had to do a journal club. And he was asking about it was a journal club about polymorphisms and exactly pharmacogenomics and which medication you should use based on it. And he wasn't familiar with the disorder that was necessarily there. And of course, you guys know that Wikipedia is not the end all and be all, that, it's a good starting place, but you should really dig a little bit further for your knowledge. And so I reminded him about online Mendelian inheritance in man, so OMIM, as a great resource on human genes and human genetic disorders. And the Broad Institute has great software tools to help you analyze genetic studies. And as far as I know, all of these are freely accessible. Okay. So if we want to talk about what are the predicted functional effects and the relative risks that a variant will alter that function, these are the different types of variants that we've talked about over the last 2 years. The non-synonymous that we talked about, evolutionary conserved, means that those tRNAs are pretty common, okay? But when you do a non-synonymous and evolutionary unconserved, if it's not conserved very well across evolution, the likelihood is that it's not going to have much of a phenotype risk to it. But if you have a radical chemical change in the coding region and a very, very low frequency, now you're going to have a relative risk of a high chain is going to be the high risk that amino acid substitution is going to change the activity of the protein that you're looking at. Okay, so you can see 4 different types of non-synonymous changes. It makes sense that if your amino acid substitution is to an amino acid that is relatively similar to the one that had been encoded previously, then perhaps a good majority of the function is going to be preserved, particularly if it's not in an active site. Okay. When we talk about insertions and deletions, they can be really disruptive, particularly if they're in a coding region. We talked about changing the reading frame in biochem and the effects of changing the reading frame on the amino acids that are encoded. So if you have a 1 or 2 base change, either an insertion or a deletion, you're throwing your sequence into a totally different coding frame. And remember that instead of having the open reading frame, when you get thrown into those other frames, reading frames, your chances are that you're going to run against, first off, you're going to change all the amino acids downstream probably. And secondly, you're going to encounter a stop codon a lot earlier than you would have expected. Okay, because the whole idea of an open reading frame is that until the transcript is completed that you need for the encoding that protein, then there are no other stop codons. But if you end up showing that you causing a frame shift, you're going to have a problem. So it could be a very low risk, especially if it's in maybe in an intron or in a non-coding region, depending upon if it's not taking out a splice site, then to a high one, a very, very high risk of a phenotypic change. So remember we talked about in a synonymous SNP, it can affect mRNA stability. It can affect splicing if perhaps this new SNP generates a cryptic splice site. So now you get an altered splicing mechanism going. Regulatory regions we talked about, and again, intron-exon boundaries, pretty much affecting splicing pretty efficiently. Intronic intergenic are unknown. It could be high, it could be low. We don't know. If this affects an important enhancer, which can be many, many bases away from, thousands of bases away from the start site of your coding region, could really disrupt the transcription of that gene and therefore affect how much of that protein is being made. So the nice thing about this table is that it kind of, it brings together all of the different types of changes that we've talked about and what the potential functional effects are. So you've got it all in one place. So when we're talking about natural variants of those of an transporter, and these are all different variants in the organic cation transporter, OCT1. And so these were identified in ethnically diverse populations, and then what the investigators did was that they put these sequences containing these polymorphisms into oocytes and had them expressed, and then tested their function. So once again, it's pretty good. You can test transporters very effectively this way. And what you can see is that if you set 100% of the transporter, the reference transporter as that's 100%, the amount of drug that was transported, MPP taken up, set at 100%, and here's your negative control of water. So nothing was put in. Now we've got these different polymorphisms. So remember, the polymorphisms sometimes are designated, in this case, these are designated by the amino acids that are changed. Okay, so so in all these cases, we have these all have amino acid changes. They're not. Well, here's a deletion mutant. The rest of these are all amino acid changes. So depending upon the number, so this is S14F, this is a change of the 14th amino acid. You can see that this change induces an increase in function. Almost all the rest of them are either neutral, so you could say that this is definitely within error bars here. But there are a couple of these changes that really decrease the efficiency of MPP uptake, excuse me. So when you have, now you have less than 50%, you're down to pretty much the same as there's being no transporter there. Now you know that then these polymorphisms are really capable of causing a great decrease in the activity of this transporter. So if we were to look at where the SNPs happen, and looking at the various types of SNPs, you see that the 5' UTR and 3' UTR don't have very many changes. And the same with the synonymous SNPs, are really not a very large percent of the SNPs that have been identified. If they're near the genes 3' or 5' ends, maybe a few more. Non-synonymous SNPs are even a little bit more common, but this is just pointing out that in this study, they found that the majority of the SNP variants that they had identified were either in intragenic regions or intronic regions. And you can think about a couple of reasons why this is. It could be that SNPs that were more detrimental didn't rise to the frequency in the population that would make them a polymorphism. If they were detrimental, it's unlikely that they would be passed on, is a big way of looking at it. But when you're looking at, you see how many intronics SNPs are identified, near 50% of the SNPs that have been identified are intronic, then you can see what some of the effects are. So for example, here we have the CYP3A5 star 1. So remember that the star 1 version of a gene, in that designation, that means that that's the canonical sequence. And typically it's the most common sequence that's been found. So this results in a transcript for CYP3A5 star 1 of, once all of these introns are removed, of having these 13 exons all put together so they encode an active CYP3A1 protein, okay? Or a CYP3A5 protein, sorry. So what could happen is if there is a SNP in the intron, this is now creating an alternative splice site. And now the transcript that is generated actually has an extra piece of DNA called 3B in it, okay? The rest of the introns are removed properly, but because you have an improper exon added in here, the likelihood is that it induces a frame shift. And you can tell that it's a frame shift because in this star 3 allele, the sequence ends up with an early stop codon. So now you have an early stop codon in exon 4. So the rest of this protein's not made, and the part of it is made is degraded, okay? So you end up with a truncated inactive CYP3A5. So if you, in the simplest form, if we had an individual and we're looking at their CYP3A5 allele, and their genotype was CYP3A5 star 1/star 3, then you would expect that there would be an intermediate level of activity overall for CYP3A5. Okay, does it make sense? Anybody have any questions? Okay. So here we are looking at this, and in this case, we're looking at the metabolism of the different drugs. So midazolam is used quite often as a probe drug. So in order for us to look at what the influence of these polymorphisms in CYP3A5 are, you can expose cells to the prodrug and then see how quickly it's metabolized. So we're looking at hepatic CYP3A mediated hydroxylation of midazolam, okay? And so it's as expected. If you have an individual who is homozygous for the star 1 allele, which means it's an active allele, then you get very robust hydroxylation of midazolam. Okay. And similarly, then, when you have an individual who's homozygous for the STAR3 allele, then you're going to have very, very little activity, as indicated by the hydroxylation. And for a person who's heterozygous, has STAR1, STAR3, now their concentrations of the hydroxylated form of midazolam are somewhere in the middle. Makes sense. So the other thing is that now if you look at, this is the mean trough tacrolimus concentrations. Once again, so now you're looking at how much has been metabolized, right? If you've got great metabolism in between, you know, the peak of when you give the drug and the trough after you give the drug, you're looking at how much of the active drug is left. Well, a lot of the drug has been already metabolized here. So with an active form of the CYP3A5 protein, now you've got very, very little left in the trough because the drug's been metabolized. Here you have a heterozygote, and you have, once again, an intermediate amount. And for the homozygous person or people who have both less active versions of the gene, now there's still a lot of drug even in the trough period. So the implications for this are that you could easily overdose somebody with the drug if they are incapable of metabolizing it properly, because instead of the normal amount being left over that you expect, which is depicted by the STAR1 on this graph, now you've got this much drug left over, and yet you're ready again to dose this person in the next 3 hours. So you would end up with an overdose for that person at that time. Okay, does that make sense? You'll let me know if it doesn't, right? Okay. So here is just another picture of the activity of, I hate trying to pronounce drugs properly, lansoprazole, because this is a proton pump inhibitor. And in this case, you're looking at the area under the curve, you're looking at the intragastric pH, and you're looking at the cure rate. So in this case, for CYP2C19, you are looking at extensive homozygous, extensive metabolizers. You're looking at heterozygotes and then poor metabolizers, okay? Once again, 3 different versions. And at this point, the area under the curve is really low for the homozygous extensive metabolizers, which is what you would expect, right? And very high for the poor metabolizers. So the effect is that you have some moderation of the intragastric pH. The extensive metabolizers, you brought up the pH some, but not as much. With the poor metabolizers, that means that the drug's around a little bit more. Now you've got a higher pH in the stomach, okay? So you've got that proton pump inhibition. What this translates into is the fact that in actuality, the poor metabolizers have better cure rate, because the poor metabolizers allow the drug to hang around a little bit longer, and therefore that pH is higher, and therefore you're going to cure the symptoms of that ulcer a whole lot better. So in this case, it doesn't always mean that the poor metabolizer is the person who has the worst outcome. In this case, those poor metabolizers had a better outcome. So what your job as a pharmacist is to think about how you use this information in recommending a dosing regimen for these different types of patients. Okay, that's why this is important for you to know. So in the real world, we've got 2 different types of enzymes that are responsible for metabolizing clinically used drugs. So for the phase 1 enzymes, we've got pretty much CYP3A4 and 5 together are responsible for metabolizing almost half of all drugs. Okay. CYP2D6 has the next big chunk. CYP2C8 and 9 has the next big chunk. There are others. But this kind of tells you that CYP3A4 and 5 are really critically important. And therefore, the polymorphisms that are present in the genes encoding these drugs, these enzymes, are really important in understanding what their effects on drug metabolism are. So the other one is now, if we look at phase 2 enzymes now, you've got a lot of different, it's split up a little bit more easily. Remember we talked about the GSTs, glutathione S transferases, and the different types, P1, M1, and T1 are all included here. And then the UGTs are also a huge percentage of the enzymes that metabolize those drugs. Okay, a lot of them are metabolized mainly by UGTs and GSTs. Okay, we'll come back and finish up with a couple of examples of how this is then used clinically. So this is our classic example of warfarin. So warfarin, remember, is dosed as a racemic mixture of S and R warfarin. And it's processed by CYP2C9 to hydroxywarfarin, which is inactive. Okay? But warfarin has an effect, the warfarin that is around, on VCORC1, and that's responsible for transforming vitamin K epoxide to vitamin K. So there's two things at play here. How much warfarin is around is dependent upon how much is hydroxylated, and then how much is there to prevent the activation of vitamin K epoxide to vitamin K. And the reason this is important is because vitamin K is responsible for activating clotting factors. Okay. So remember that when a patient is on warfarin, it's because we want to prevent clotting. And the issue with warfarin is that the therapeutic index is very, very tight. So when a patient is first put on warfarin, quite often they initially come in several times a week and then processed to once a week, then to once a month, as the pharmacist is titrating the levels of the drug that they're getting. Okay. So the recommended starting dosages can change depending upon the genotype of the patient. If the genotype is known for these two, you can start off a lot closer to getting where you need to be with that patient. And this gets really critical. We talked about the fact that people who have jobs where they cannot take off to come into clinic to get their INRs checked, they may have to go a very, very long way to get to the clinic, and transportation may be an issue. Typically, rural areas are affected quite often. So the whole idea behind genotyping for warfarin dosing is that you can start off closer to the correct dose to reach the therapeutic index that you need for that patient. So this graph on the right is a combination of looking at the CYP2C9 genotype and the VCORC1 genotype to see how they combine to come up with a recommended starting dose in milligrams. Okay. So holding one thing in common, the VCORC1 homozygous variant BB, now you can see that if you have the CYP2C9 variant that is homozygous for A, you need a lot higher starting dose than you normally would. Okay, normal starting dose would be 2 or 3 mg per day. And then depending upon the other 2C9 genotype, you could perhaps start at 4 mg per day. Okay. Now say that you have, let's take the other extreme, and you've got a patient homozygous for the VCORC1 AA genotype. You can see that, and with the CYP2C, various CYP2C9 genotypes, you would start off at a lot lower, at 3 or 2 mg per day. Okay, and you can see that there's overlap. So depending upon the combination of the two, in this case, you can see that the CYP2C9 genotype being homozygous for B and the homozygous variant BB for the VCOR, you're now at the equivalent doses of a person who is at the opposite end of the spectrum for 2C9. But them having a heterozygous variant here with B brings them up and increases the dose that is needed. Okay, so you can see that there's overlap depending upon which allele is there most off, or which is more active in the process. Okay. So the next example is codeine and CYP2D6. And I think I told you about my mom. My mom is one of those people who must have been missing a copy of the gene, but basically codeine didn't do anything for her pain at all, because it's a prodrug, and therefore it needs CYP2D6 in order to be activated and metabolized into morphine, because morphine is the active form. So this is a metabolizing enzyme in the liver, and there are a significant proportion of Caucasians missing a copy. So this must have been what happened to mom, and she must have passed that gene down to my brother and sister, because codeine doesn't work for either one of them. Works fine for me, so I'm happy. But so now when talking about SNPs and drug development, some SNPs are associated with absorbance and clearance of different therapeutic agents, which can be make them either effective or ineffective. And it really depends on if we're talking about drugs and prodrugs, okay? And this is where adverse reactions and personalized medicine come into play. So you want a drug that's effective and doesn't have any adverse reactions. So if it's ineffective, it's like somebody taking candy. It's not going to help them at all. And adverse reactions can be severe enough to kill someone. So this is the whole idea behind the influence of personalized medicine on drug development. Remember, we talked about frequencies. So one of the things to be aware of is the ethnic background of the patients that you're looking at. So you can see that in Caucasians here, anywhere, depending upon the study, between 12 and 21% have, and another additional 2 to 7% have CYP2D6 alleles that make them poor metabolizers. And then by the same token, you have CYP2D6 people who have ultra rapid metabolism. Two different problems. But if you have an Asian patient, perhaps not as common to have at all, to have a poor metabolizer or an ultra rapid metabolizer. For Blacks from Africa, very, very common to have an ultra rapid metabolizing genotype. Okay. And so here's an example. If you have acetylation polymorphisms, this is, remember, this is one of those that conjugate substrates with more water-soluble moiety, so that enhances excretion. So think about it. Does that make it a phase 1 or a phase 2 enzyme?
Phase 2.
Very good. Yeah. So in this case, you have people who are fast acetylators, and so some of the consequences to some of these drugs and some of the carcinogens that we are exposed to in cigarette smoke and perhaps an industrial setting, you could end up with a hypersensitivity to sulfonamides. You could end up with toxicity. You could end up with hydralazine-induced lupus or neurotoxicity, which is even more of an issue. All of these are pretty issues. But you can see that if you have a fascicetylator, it depends on the population that you're examining as to what are the potential percentage of patients that you're seeing that you're going to need to worry about this. We have different cancer treatments where this is just here an FYI. The idea that these are the 4 biggest cancer drugs for which we know that variants in genes that are used to process them can end up with having quite a lot of adverse phenotypes. So myelosuppression, and the FDA label recommends genotyping. Neutropenia, again, recommending genotyping. Tamoxifen metabolism. So if you cannot process tamoxifen to endoxifen, so tamoxifen is a prodrug, and it gets metabolized to endoxifen, which is what is going to affect your ability to progress, or the propensity of your cancer to progress. Pretty much, if you are inhibiting CYP2D6, your drug is, you might as well not take it, because it's not going to be processed. It's like taking codeine when you don't have CYP2D6 activity. You're not going to get any efficacy out of the drug. It's just going to be wasting your time and wasting your money, and subjecting the patient to potential progression of their disease or metastasis. Okay. So there are purine analogs that are used to treat different leukemias, some autoimmune disease, IBD, and are also used to inhibit the immune response after a transplant. Okay. And if you remember from way back in biochemistry, when we talked about nucleic acid synthesis, these 3 drugs interfere with nucleic acid synthesis. But the therapeutic index is limited by the myelosuppression. So in some cases, you want immune suppression, but you don't want so much immune suppression that your patient is going to die of the next infectious disease that blows through the building. Okay. But these 3 drugs, and you can see by their similarity to different building blocks in DNA, you can see that how these would interfere then with nucleic acid synthesis. So thiopurine S-methyltransferase is the phase 2 drug that catalyzes these thiopurine drugs. And it's part of that inactivation pathway. So if you have homozygotes for a non-functional allele, then you're going to get a buildup of the drug in the body, and you're going to end up with hematopoietic toxicity. So it doesn't mean that you can't use the drug. You just have to be very careful about the dose. So if you are not, if you're not excreting the drug, if you don't have, we talk about ADME, if you don't have excretion, you're going to get a buildup. And so you can really deal with that by reducing the dose tremendously, because it doesn't mean that there's no excretion. It just means that it's really, really slow. There are other pathways that are going to be used, but they're not as robust for this particular drug. And the key here is that heterozygotes could also be a greater risk, because the heterozygotes could have some inactivation going on, but not as much as you would expect for the canonical patient. So you may have to monitor heterozygotes and reduce their drug dosage regimen as well. Okay. So here is a way of thinking about it, and this is a really good example, is that if you have any one of the star 2, star 3A, star 3C alleles, you're going to have pretty much a deficient amount of TPMT around. Okay. So you can see that the activity is really, really low. The percent of drug that is being processed is very, very low when you have any of those star 2 or 3 alleles. Whereas with a wild type, now you can have a decent amount of activity and a decent amount of excretion. So then the idea here is looking at individualized dosing. So if you were to dose everybody the same, you're going to end up with a poor outcome. When you actually adjust the dosing given the genotype, you can see that a person who has this MM allele, this minimal allele down here that barely has any activity, you would give a much, much smaller dose. So this is mgs per meter square per week, and we're down in the 20 range as opposed to 250. Whereas a person who's heterozygous now, you may be up at 200. And a person with a wild type, you may have somewhere around 250 as your dosing regimen. So a big problem with cancer treatment is that because of these individual differences, a patient could be undertreated if they are treated with a drug and the drug is the active form. And if it's metabolized really quickly, then it doesn't have enough time to kill the cancer cells. If the drug is metabolized really slowly, or they're subjected, it hangs around their system too long, now they end up building up to toxic levels. And so the classic example of this is 5-FU metabolism. There are 2 genes that come into play with 5-FU metabolism that are responsible for a lot of the toxicity. And pretty much it is recommended that looking for, in particular, the DPD deficiency is be considered. So here's the example in cancer treatment. In the case of a normal person, they are given a dose, and around 50% of the drug, 15% of the drug is active, right? So when it binds to the enzyme, it's going to take out about half of this enzyme here. And then now you're going to have an effective therapeutic dose. You're killing the cancer cells and not killing too many of the healthy cells. This is when you have the proper amount of the thymidine synthase enzyme, okay? If you have a DPD deficiency, and DPD is encoded by TPYD, you've got lower amounts of the enzyme available. So you've got every single enzyme site is occupied by the drug. The drug is killing all of the enzyme and therefore killing both cancer cells and healthy cells. So you're inhibiting DNA synthesis and you've got an extreme adverse event going on. So then let's look at the other player here. We're looking at the TS enzyme. In this case, a person is given, TS is given the same amount of drug. If they were given the same amount of drug, now all of the sites are occupied. And it's not because there's too much drug, it's because there's not enough synthase. And you end up with the same problem, where you end up killing both cancer cells and healthy cells because you're inhibiting all of their DNA synthesis. So then you got to think about what you would do with the dosage in each one of these cases, okay? What would you recommend to the physician who's in charge of coming up with the dose? When you think about that. Because you've got 2 different situations here. You've got a situation where you have binding to an enzyme, you have lower amounts of enzyme and higher levels of drug, and now you've got a case where you don't have enough active drug, but you don't have enough synthetase around. Okay. So here's a bunch more of drugs where for cancer treatment in which we really have an issue with knowing what the genotype is and knowing whether or not these are the proper drugs for those patients. Okay. So, does anybody have any questions?
I have a question for you, Liz.
Sure.
So I guess what's kind of the info you want us to take from most of these slides? The overall theme, I guess.
Yeah, I'm not going to ask you picky details. What I want you to understand is the difference in how you would approach dosing if you're giving somebody a prodrug and they have some polymorphisms that alter the efficacy of that enzyme, or if
Okay.
you're giving somebody a drug. Okay.
Gotcha.
So that kind of thing. The difference between what would you do in the case like somebody has an inactive enzyme? What would you do in the case of giving somebody a prodrug? And what would be the consequences versus what if you gave them a drug, what would be the consequences?
Well, if they don't have an enzyme to convert the drug, then I wouldn't give them the drug at all.
Right, there you go. Find an alternative or
So I got it.
find a drug that uses an alternative pathway, right?
Okay, or yeah, that too, I guess.
Yep.
Cool.
Yeah, I don't expect you to memorize these different types of drugs and the different type. What I will try to do with the questions is give you enough information that if you are aware of the concepts, you're going to be able to successfully figure out what you should be doing in that particular case.
All righty, thank you.
No memorization. You know me, I don't like memorization.
Yeah, sure.
Oh, I'll remember that, Tristan.
Thank
Okay,
you.
sure. Any other questions?
So for the example cancer treatment one, for the one with the last one, the TS deficiency, wouldn't you just have to change the drug? Because I mean, even if you lower the dose without enough enzyme, it seems like it's not going to provide proper treatment no matter how low the dose is.
Yeah, exactly. But these are the kinds of things that people are in the past. It's been trial and error. You know, you give somebody a drug and you figure out that it either works or it doesn't, or it kills them. We actually when I was training at MD Anderson, we used to call it in developmental therapeutics department. We used to call it detrimental therapeutics because it seemed like half the time they tested something and the outcomes were not that great. So yeah, but it shows why it's important. And in this case, you've got two different possibilities of either not being able to process to have enough drug, or not being able to have, or ending up with, no matter how little you process, having an overdose, essentially.
Thank you.
Sure.
So Liz, on that one, for like that 5-FU with that TS deficiency, is there would be no other drug that we'd be able to like then give them to try to increase that TS enzyme or like block it to where they could have more? Is it just that like there's no way to get them to have more of that TS enzyme?
Yeah, that's the key right there, is that if there's, you know, it's not, they've got enough to obviously grow to adulthood, and they've got enough, you know, DNA synthesis going on that they're healthy. But I think that it's more of a case that you would choose a different drug.
And so when most of this stuff happens, do we find out like more so now, would they just switch the drug, or like with any of this stuff? Because I guess like when we mix chemo, like, I mean, I rarely see like a huge decrease in the amount of doses that's being given for a patient. Like either they're on it on their same dose, or like then they change therapies. I don't see very often in with me working hospital and chemo side, like I don't see very often where they change their 5FU dose very often. It's either like they're on it for a little while and then they stop and change therapies completely. Is that just because so
It's
many
because
people just don't do the treatments to find out what's wrong?
it's because it's easier.
Okay.
You know, it's you know, if you basically, if it's not working or if they're having really bad side effects, the simplest thing is to move them. I mean, people still use 5FU because it's so effective when it does work. It's great.
Yeah.
But the problem is anytime somebody has an issue, it's kind of not worth the time and the cost to figure out why. Because as you say, they're not going to change the dose. They're just going to go ahead and switch.
Gotcha.
Any other questions? All right. Wish I could see you all, but hopefully sometime in the future. You guys take care.
Have a good day.
You too. Bye-bye.
Thanks, Liz.
Thanks.
Bye, Liz.
Bye-bye.